Okay, here we're talking about writing assignment 1. And as it says here, WA1 is where we describe your mutant. So you have your mutant, you have the letter anyhow that you were given, and that is a description of the mutant phenotype that you are describing your fly of, and it's a particular mutation in the genes, of course, but you see that expressed as a phenotype. And so you're going to write about the phenotypic characteristics, and slowly we're going to add to that so that you know more background behind it. And you're going to begin writing this paper that we're going to call WA5 in the end, but it's WA1 right now. Right now it's very basic, and you're just going to get started. The whole idea here is to get some general idea about your mutant for this paper and to get you started writing it correctly in the right format, so that you have the right spacing, format, justification, get rid of all your spelling errors and stuff like that early so that you can begin to add to it. And by the time you get to WA5, you have a really nice paper and you've got all the organizational stuff under your belt. You know how to do citations, that sort of thing by the time you get to the end, where at the end you just have to worry about, adding some more technical detail. You already know how to, what do you want to call it, write the paper. All right, so in the example, or I'm going to show you an example, I should say, you're going to see a final paper, just so you can see what one looks like. But before we do that, let's go through just what things count as you begin to write this paper. The font, the spacing, the margins, the justification, all of that is based on the descriptions in the other part of the rubric that Dr. O'Brien has given you. And you can see you get 2 points for each of these things, including grammar and spelling. You got to check for spelling. Now, the length needs to be 1 to 1.5 pages of text, 2 pages max. So you can go all the way to 2 pages for this. So this is not a huge assignment. It's just 2 pages maximum. Don't go over. We'll count off for going over. So 1 page for a figure and a figure legend, and 1 page of references. So here, this is a text. That's 2 pages. Then you're going to have to create a figure and a figure legend. We'll go over that. And then you have 1 page of references. That is up to 10. Now, you also have to use the correct nomenclature. You have to, the names and the symbols. Do they need to be bolded? Do they need to be capitalized? Do they need to be italicized? You have to learn the rules for using genetic nomenclature, like a gene or a protein. How do you draw those? How do you write those? And how do you distinguish between one thing or another? What is it? What is the gene that causes the F mutation? All right, things like that. You're going to have to say, well, the gene is this, the protein it makes is this, and when you say those things, you need to capitalize, for instance, maybe the protein, something like that. You may have to italicize the allele, things like that. You have to know those things and put them into your paper correctly. Now, what you're going to be also graded on is contrast your phenotype to a wild type. Now, the wild type, of course, is a fly that is it's essentially the most common alleles, the ones that you see in nature, in the typical fly flying around. Basically, you know what the wild type is by looking at a standard Drosophila, and that would be often that's represented in the literature as a plus, or maybe as a WT for wild type or plus. Those are all standard methods for describing the wild type fly and the phenotype that is the wild type. Anything that's not wild type is a mutant, like F or E or BW, something like that. Those are the non-wild types, and you have to distinguish between the wild type and these mutants. Phenotypic similarities. So in other words, sometimes your mutant is going to be different. There might be difference between here, males and females. Maybe the males are all wild type and the females are all mutants, or vice versa, something like that. You need to notice those things or read about those things. We're not going to be able to do them directly. You're not going to be able to breed flies, but you will be able to do it in a computer simulation. Let's see. You do need to make one figure, and I'll show you a figure. That's essentially a picture, and then you write a legend underneath it, and that's worth a certain number of points. You need to find at least 4 references in this first part. Might not be a bad idea to get more than 4. Description could be used, yes, and then you have a description, and the idea is to write it well enough that you can use it in WA5. So here are some additional rules that Dr. O'Brien has set down. But within these rules, you have my interpretation of these, and you need to basically talk to me when you go beyond, when you have a question that goes beyond anything listed here. So these are going to be your points and how you're graded. Beyond this, though, you have to come to me. So you look here, do what it says here, but then if you have more questions, like what is conciseness, things like that, and there will be a whole video on what is concise. So at this point, we're just talking about, here are the basic rules. Read through this, get a feel for it, and let me show you some things first. Let's go here to Flybase. Let's see if we can go to Flybase. So you click here, and what you do here for Flybase to learn a little bit about your mutant is, notice I just clicked on that that link there within the document. And what we're going to do, it says jump to gene. Well, there is a gene called BW, brown, if that's one we're not using. So I can go here, and we'll wait just a second, and it's going to find out all kinds of stuff about brown. So as you can see, D-mel stands for Drosophila melanogaster. BW is the mutant within it. That's the annotation symbol for brown. All right, so we have, what is it? Brown encodes for a member of the traffic ATPase or ABC family of membrane transporters. It is believed to I have a hard time saying that, heterodimerize with the product of W. BW and W are necessary to import guanine, all right, a precursor for the red pteridine pigments into pigment cells, in other words, into the pigment cells of the eyes, the Malpighian tubules, and testes sheath. In other words, it's a pigment that is produced, and if BW and W don't work right, then you can't get the red pigment to the right places, so things that are red on the fly don't show up as red, or maybe they don't show up as red at all. So therefore you end up maybe with brown. Things end up being brown instead of red. So that's kind of the idea here of what the BW mutant is. And so you will need to understand your fly. Here is a general description. Of course, I explained it. It's, you read this, you're going like, I have no idea what that means. So I've described it a little. You would have to go into the literature and find out about this. And these things are in Wikipedia. I mean, there are internet sites that describe all of this. You can just do a Google search and find many sites that would tell you about this particular mutant, say BW. Now let's see what happens when we click on BW. It's going to take us somewhere else in Flybase, and actually takes us to the gene here, and various versions of brown. Now that's confusing to some people, but there are different types of brown and brown related genes that can lead to this mutation, that can lead to this phenotype. This is useful, maybe not right away, but this might be useful to you. Try to remember it. This might be useful at a later time in the paper. So remember that if you can find that for your mutant. All right, and so will the FASTA. All of these things will be useful later on. But let's scroll down here to summaries. This should help you quite a bit. You can read about various versions of BW. You can find out more about it. You can find out the protein, biological processes that it affects. Everything is here for you writing your paper. You just have to be able to read it and figure out what it means. All right, now we scroll down. That was the summaries. There's a gene model and gene products. All right, now this isn't going to make sense to you yet, but it will in the future when we get to the bioinformatics. expression data. All right. Alleles, different versions. There are classical alleles, transgenic, deletions. Those might be useful to look at some point towards the end of your paper. So remember those things. There are versions of BW, many of them. They've found many ways to produce this phenotype. The phenotype is what it looks like, but that phenotype might have several different mutations. There might be several genetic mutations that could lead to that phenotype. Orthologs and paralogs, don't worry about those right this minute. You might want to talk about human diseases, that you might study this gene in order to understand something similar in humans. If you want to do that, you would click here. Pathways, certainly something you should look at. Now this is more of if you were going to do the experiments on your own, what kind of things would you need? There are a list of references here that might help you complete your paper. I would certainly at some point look at this, and in fact there are some references here that will tell you everything you need to know for WA1. You're just going to have to go find them and start reading. All right, so this is a general idea of how to do it. Flybase is a database of fly mutations, fly genes, fly proteins. Everything fly is here at Flybase, especially if it has to do with genetics or bioinformatics. So it's a very central place for us in this class. All right, so this is where these little drop downs here, especially down here in the references, are what you need to complete WA1. All the information you need should be located at least somewhere around here. There are easier ways to get this. Literally, you can, with the right search in Google, you can come up with things that are actually probably easier to read and more informative than what you would find here. But this is a place to get direct references, and that's one of the things you need. You need to find some references, you need to read through these, and see if you can't figure out some things. It'll give you an idea of how to write a technical paper as well. Of course, don't plagiarize. But otherwise, this is a good place to go to start on WA1. Once you have all of the like rules down, you get a pretty good idea of what we're asking of you. Then you would maybe go here, maybe do a Google search, start kind of getting your ideas for what you want to write, and then you would come back. And here is I'm going to open something else up for you. Let me find. Let's see. Well, I thought I had it here. Here we go, example paper. So what I want to do is I want to show you an example paper. Now this paper is from several years ago, and this is WA5. So this is what the final paper that you're going to be writing should look pretty close to. This would have more, there was more to it than we are asking you, so it certainly doesn't have to be 61 pages long, but let's start. So here's your title page. So it needs to it'll look something like this. All right, have this type of information. You'll have a heading only on the first page. You don't have a heading after that, just the first page. Don't forget that. And then you do the abstract. So the abstract will be one of the early requirements where you have to write an abstract here, or I guess actually it's one of the later ones, but the idea is this is just an introduction of your entire paper. So you should read that. Then the introduction. A lot of the things you're writing in WA1 will go in the introduction. All right, and you can see that that's several pages. Notice how the references are done. All right. Notice all of these what? Italicized gene. See the italicized and the abbreviations. All right, make sure you understand how to do that. So notice Drosophila melanogaster. Okay. The first time you use the term, you use it like this. Later on, if you want to use it, you have to put the abbreviation in parentheses. I don't think they did that here in this paper. Let's see. DMEL. So they see this is the abbreviation, and they didn't put it in parentheses after this, so that would have been counted off. That's probably what this is. So see, black, the first time you use it, ebony, E. When you use them, you need to, the first time you use it, you need to put the abbreviation in parentheses. Don't forget that. Okay, so you're going on, and yeah, they did not do that here. See, they used the abbreviation here, but they didn't put the abbreviation in parentheses the first time they used it, so that would have been counted off. All right, so make sure you recognize the way that these are referenced. Notice the distance here. Several things here. These little things are places I counted off for something. But you can see that this is the kind of idea. Then you have materials and methods, and then you talk about specific things materials or methods that you used. All right, this part was very extensive in the old papers we wrote before COVID. This part will be smaller in your paper because we don't have to worry about all these tools and things, agarose gels, we do a little of that, but we don't do as much as we did before. All right, so let's scroll down. So the next piece, molecular basis. What do we got? Discussion. I think we skipped a section there, but just showing you, trying to scroll through this quickly to show you what a paper actually looks like. And let's get down here to the references. So this is the format for your references, and something very similar. They should get their own page. All right, so when you finish the paper, you skip to the end and start a new page, call it references, 1, 2, 3, 4, 5, however many you're going to put in there. This person only put 11. I suspect they got counted off a little bit for that. Should have been at least 12. Then you do tables and figures. Also, skip to the end of references. Tables and figures should start on their own page at the end. All right, now this came after the references. So if you have tables, you have to put a description underneath each one. So here is your table, and here is a description. Now in WA1, you're going to make a figure. This would be a different type of figure, a thing, but most of our figures are going to be, that for WA1 that you're looking at, is going to be a picture. So let's scroll down to a picture so you can see what a picture looks like. So this is a good picture. They had 1, 2, 3, 4, 5, 6, and this is figure 7. All right, they give it a title, and basically it's bolded. And then they have A, B, C, D, E, F, so they labeled them, and they described A, B, C, D, F. Now you might also put in little arrows and describe what the arrows are. Here we have a gel. You guys aren't going to be doing much of that. You will be doing bioinformatics, which will leave this. So you'll have more of this stuff. And that's it. The idea here is you have pictures. All right, you have figures, tables, and they go at the very end. So in WA1, you're going to have to make a figure, probably find a picture of your fly, compare it to a wild type fly, and then you also have to put in a few references, and then you have to write something in some type of description of your phenotype based on those references that you got. So make sure you put in on the first page the heading, give it a title. Kind of, you don't have to do anything this fancy on the first one. It's really just do it. You don't have an abstract on the WA1. WA1 is just a description. It's not a full paper. But get in the habit of doing things, formatting it right, and especially on this first paper, get the italicized and the abbreviations, that sort of thing. Find out what the rules are, and get that corrected so that you get into the habit for it. Now when you reference one of those figures, you talk about the figures in the paper, you need to put them here. So figure 7b, that would have been one of those flies we talked about. So figure 7, I believe, was the pictures of the flies, and the second picture would have been B. So this is, see, the dark color is especially seen on the scutum of the scutellum, the wings, the veins, and the wings on the abdominal tergites. All right, and of course you probably don't know what any of those things are, but the idea here is figure 7 all of those things are going to be there. All right, that's what it's telling you. All right, so keep in mind this is kind of an example of what we're looking for. This, of course, was a completed WA5 back when it was longer. Yours is probably going to be much shorter than this, but maybe we'll go back to it, but I don't think we will during COVID. All right, so just wanted to show you a finished paper. Read through Dr. O'Brien's descriptions and everything. That should help you with what I just showed you. And we'll make this short. There'll be more descriptions besides this one. This one is just getting you started, showing you kind of where you can go, how to get some information about your mutant.